Generally, when preparing a PCR reaction, there aren't too many components involved. The hardest part really about preparing a PCR reaction is these teeny tiny volumes that you will be pipetting. So for this, we're going to use a different pipetter. This one is a P20, which means it goes from, we can pipette 2 microliters up to 20 microliters. So these are going to be very small volumes. And then the other difficulty is working with this tiny little tube, right? Comparing that to a normal microcentrifuge tube, we got a little one right here. So for this, using a fine pen is much easier to label. So I'm going to label with my initials. I'm going to require my old man glasses so I can see. I want to make sure I don't miss the tube. I have my DNA that I prepped, right, by taking a colony and boiling it. And then I have this thing here called a PCR mix. Now in the PCR mix, what we have is we have a buffer, of course, for things to be okay, working properly, right pH, right salt, et cetera, et cetera, et cetera. We have our primers, which we need to amplify that 16S gene, 16S RNA gene. And then we also have the polymerase, the enzyme that's actually going to do the amplifying. And that's pretty much it. Water then to bring the rest up to volume. And if you look at it, it looks just like water. So just be aware when you're pipetting things and grabbing tubes, you know, everything's going to look the same. And it's all going to look like water. So just make sure that you know what tubes you're getting it from. And generally the workflow I like to do is I like to keep things on my left. And then as I work with it, I pick it up. And when I'm done with it, I move it to, did I say my left? I meant my right. And then after I'm done with it, I'll pick it up and I'll move it to my left. And if you have many, many tubes, you do what's called a tube walk, right? So you simply pick up and then you move it like that. And then you know which ones you've done and which ones you've still to work with. In this case, I only have 2. So I'm going to just keep them on the right here and just move them to the left when I'm done. And then I have 1 PCR tube. Of course, if you have many, I would also start with all my PCR tubes to my right. And then when I'm done loading them, I will move them to the left. And sometimes what I'll do is if I have to put multiple things in, I'll put it in, move it to the left. And then when I put in my second thing, I'll pick that up and then move it to the right. And just keep working back and forth so then I know what it's done, what isn't. For this, everything is going to be pre-made, pre-mixed. So it's very simple. All we need to do is take 18 microliters of our PCR mix, put that into a tube. 2 microliters of our, we're going to call it DNA, right, our cells boiled, and put that into a tube, and then that's pretty much it. Again, the challenge is going to be trying to get these small volumes in there. So let's start off by setting our pipetter. This one's a little different. Right? Make sure this switch is flipped down to the unlock. Right? That goes down, and then we're going to rotate this until the label reads, or the dial reads, 180, and that's 18 microliters. So the black numbers are microliters. The red number is 0.1, or I guess 0.1 of a microliter. So this is 18.0 microliters, and then we're going to lock. Okay? Operation is same as before. First stop, second stop. Okay? And then smaller tips. So I'm going to open this up. Nice and snug. And then I'm going to take my PCR mix, 18 microliters of that. First stop. Grab my PCR mix. All right, and then to the left, I'm going to grab my PCR tube. This one's going to be a little bit more challenging to open. Careful not to touch your tip anywhere. So usually I'll hold it like this. Open, and then put your tube or tip down to the close to the bottom. How well can we see that? Close to the bottom, and then gently squeeze, moving your pipette tip out. Second stop, make sure you're hanging on. Pull it out. Then you can let go. And because I'm done with that, I'm going to move that to the left of my rack. Now I need to put my DNA in there. Okay, so right now what we have in here is our polymerase, our buffers, our primers. All right, so we're going to leave that on the left here. Now I'm going to stick my DNA in there, and that's going to be 2 microliters. So once again, make sure we got to unlock. I'm going to set the dial now to 2.0. So I'm going to fast forward this part in my video. Remember, black is microliters, so that's now 2 microliters, so 2.0 microliters. And then once again, I'm going to lock it. Now this is going to be a very small first stop, just like that, okay? And then second stop. So that second stop is much, much larger than our first stop. So just like that, that's all we need, okay? Once again, get have a tip. Here's my DNA. Open. First stop. teeny tiny little amount. I don't know how well that shows up in video, but just a small amount. That's all that it takes. Move it to the left. I'm going to take out my PCR tube, open that. And then here now what I want to do is I'm going to stick my pipette tip just above the surface, not into the liquid, and let the two bubbles make contact. Second stop, all the way out. Spit that out. Make sure it snaps. And then this goes into the thermocycler, right? Ready for our PCR reaction. Now be careful, these are thin wall PCR tubes, right? So if you push too hard, let's see if I can get it to do it. Nope, maybe not. But you can squeeze and break these things, right? So when you close these lids, just hold it gently but firmly and then close. Don't push too hard, right? Because you can snap these things if you hit it at the wrong angles and the wrong pressure. And that's it. This goes into the machine. A couple hours later, PCR reaction will be done.